Good afternoon. We'll get started here on today's webinar. My name is Derek Pizarro. I'm with AST Environmental. We're one of the RPI group members. I'll be emceeing today's presentation by Dr. Ed Winter with RPI. And just a few housekeeping items. We do expect this to take about 35 minutes or so. There's also a chat function and a Q&A function. You can queue up and ask questions there. We'll get to those at the end of the presentation, but feel free to enter those at any time, and then we'll do a question and answer session at the conclusion of the talk. And with that, Ed, I'll hand it over to you.
Okay, I appreciate you guys showing up today. This presentation will be kind of a voyage of discovery. So let the information kind of wash over you and see where you are at the end. We're going to focus on the obligatory dehalogenator, Dihalocartes McCarty. And what I'm going to try to show you, and see if you believe it or not, is that you probably don't need to bioaugment with dihalocoides in most circumstances. Added a little picture of it right there in front of you. And as a fun fact, dihalocoides is about 1/10 the size of the microbes you'd normally look at in petroleum hydrocarbon degradation. It's a small thing, but it kind of helps you think of how dechlorination is very different from the breaking down of hydrocarbons. So let's get started. Okay, in the biotic reduction of PCE, enzymes catalyze each step. The absence of a particular enzyme will result in the stalling at that point. So most of you are fairly familiar with the problem of daughter compounds, and those come from having a number of microbes which are facultative dehalogenators, such as I have listed in that gray box with the arrows pointing up. So you have the enzyme, say PCEA, and that'll get you to DHC, and that's where you stop. That's why in 87, when a dehalogenating culture that would drive PCE all the way to ethene and ethane was discovered, people were pretty excited. And that's our Dihalocoides. Now, the species name has changed in the interim time, but that's not really a concern at present. So here's the genes that help transfer from one molecule to the next. Some of the enzymes will catalyze more than one step. There are several bacteria that can take you to DHC. The ones I've highlighted in gray are the ones that are most well known. So most of you have heard about dichloromonas, desulfibrio, and Smithella as an example. So let's think about, well, chlorinated compounds. Where did they come from? Are they only man-made? So reductive dehalogenation itself is probably an old process that existed long before the Earth appeared. You can see perchlorate in the caliche salt deposits of the Atacama Desert in Chile. These are atmospheric deposition. There's also chloroorganic compounds in cold seeps in the South China Sea and in our own Eastern Gulf of Mexico. These are also not of human origin. I have listed here some naturally occurring chloroorganic compounds like chloromethane, chloroform, etc. So the picture I have, in case you're curious, is from the South China Sea, and you have here a bunch of clams. So I would think that reductive dechlorination is a central rather than a supplemental process in these deep sea areas, and that seems to be the consensus among scientists. These compounds occur widely. So if you look in media, you can find chlorinated compounds in aqueous media, oceans, freshwater. You can find them in terrestrial media, forest, farms, rice paddies, etc. You can even find them on animal samples. And geographically, they've been found pretty much everywhere where people have looked. And this specific picture that I'm showing you now is for Dihalocoides, which is an obligatory dehalogenator. So it's a little bit different than what I showed you before, and that I was showing you both facultative and obligatory. The interesting thing about obligatory is the chlorinated compound has to be present, as the word indicates. Otherwise, the microbe could not be present. The ability to respire organohalides is also present across many phyla. Most of you are familiar with chloroflexi, the one that I have at the bottom of the list in bold, because that's the phyla that includes Dihalocoides. But firmicutes for Dihalobacter and Desulfobacterum are also important in dehalogenation, as are the delta bacteria at the top of the list, for which Geobacter is certainly well known. So I kind of shifted in moving back and forth between Dihalocoides and the organohalide reducing bacteria. But organohalide reducing bacteria, I'm going to contend, will arise if the conditions are correct, and specifically Dihalocoides will arise if the conditions are correct. So the arguments that I have for that are more general, but we have the existence of these biochemical chloride cycles. We have a diversity in nature of organochloride compounds. You have the microbes that do dehalogenation are widely distributed in both media and geography, and the existence of organohalide respiring bacteria across many phyla, all suggesting that organohalide respiring bacteria evolved before the manufacturing of man-made chemicals. So perhaps bioaugmentation is not typically necessary. Maybe all we need is the right stimulus. So that's what I'm proposing. And I have seen that with remediation using the CAT 100 product. So I'm going to take a couple slides just to tell you what that is. Otherwise, there's no context to things I'll show you following. So CAT 100, its key elements. It's an activated carbon impregnated with metallic iron. It has BOS 100 as its base. So BOS 100 is the carbon with metallic iron impregnated. We add to that complex carbohydrates, such as food-grade starch. We add one set of microbes that is for degrading the COCs, the chloroorganic compounds, and a second set of microbes that degrade the carbohydrate, fermented to be particular. This gives you a kind of combined remedy in one technology. So you have both the biotic and the abiotic pathways operative. Now, I need to make this point, and that is that if you look at the picture on the right, this is about as good as you can get. You have a zero-valent nano iron that is on the activated carbon. That is not what BOS 100 is. It is actually in, and this picture is showing you the iron within the carbon. It is a very different product. Nothing else on the market is like it. What's important to think about in this is that the iron and the carbon are both conductors, and they are electrically and chemically one item in the BOS 100 compound. This is made by heating the iron and the carbon to 850 degrees C so that they essentially become one thing. Okay, so now what I want to do is look at a site where we had injected both cat and boss on the same site. Now the column to your left is side gradient to the cat 100. So this is just a cat 100 injection. The column in the middle, side gradient to the cat 100, it's just a boss 100 injection there on your left. In the center, you have a cat 100 PBR, and the microbes are arranged according to what's present in greatest numbers in the cat injection. And then to the far right, you have down gradient of the cat injection. You have another boss 100 injection. What this is intending to show you, or what I'm intending to show you, is that on the left side, you do not see Dihalocoides macarte being present in the boss 100 injection. In fact, you see that most of the microbes are not shared. In the cat 100 injection, you see Dihalocoides macarte, and then down gradient in the boss 100 injection, you start to see it also because the PBR, the cat PBR, influenced the boss 100 down gradient. What I want you to kind of take from this is the contention that the additional matter added to the cat 100 is essentially turning the down gradient boss 100 into cat 100, and that's why you're kind of seeing this decay Dihalocoides macarte occur when it did not in the boss 100 injection alone, all on the same site. Now, this is another site, and this is a little bit different kind of data. But nevertheless, I want you to look at the blue arrow. In fact, I'll put the little red circle around the microbes themselves, and look at the blue arrow on your left. It points towards a small blue column where you have about 100 microbes. So the Dihalocoides in this particular site was already present before injection. So prior to cat injection, it was present. In 16, after the injection, you'll see the Dihalocoides increase, and it continues to increase and stays above 10 to the fourth cells per mil throughout the period from 16 to 21 in which this chart represents. You also see Dihalobacter go up. You see Desulformonas also. And you see the appropriate enzymes right here for degrading post-DCE. Total U bacteria, which is the last column, also stay high. So this isn't actually the strongest argument because Dihalocoides was already present, but it does clearly indicate that the injection changed by increasing the Dihalocoides, Dihalobacter, et cetera, and increasing the appropriate enzymes and increasing total U bacteria. Now, at this point, I'll actually show you some of the chemical data. I won't show you any more of that during this presentation, but it would seem remiss not to show you. So on this same particular site, the red line is TCE. The vertical black line is where we start full-scale injection. The double vertical line is where we end full-scale injection. So it's not surprising that the TCE dives immediately, because you could argue that it would do that from adsorption anyway. But that's probably not the only process, because you see that both the DCE and vinyl chloride go up. So the daughters from degradation start jumping around, but ethylene climbs more consistently up. And this is kind of the cat pattern. Some sites you'll see the pattern, you won't see as much variability, but this is a pretty high concentration site, as you can see. Cold total chloride generation is also consistent over time. So if I were to argue that the halicoides will appear if you have the right environment, what would we need for that to happen? Well, the first thing I want to talk about is that the halicoides McCartae, all the obligatory dehalogenators that I'm aware of, for which we're just using D. McCartae as a model, require exogenous coronoids. Now, vitamin B12 is an example. And so a lot of times you'll hear people say you need B12, and that's shorthand, but it's correct. As far as we know, known D. McCartae strains cannot synthesize coronoids de novo. In other words, they can't make them themselves. The microbial community must synthesize these coronoids. D. McCartae 195 is capable of remodeling coronoids if they're given by the larger microbial community. So no B12, no D. McCartae. So now I'm going to spend a little more time than I probably should just looking at the reductive dehalogenase as a molecule, as a series of molecules, as a physiological structure in the cell membrane. I won't make it, it's not going to be difficult or anything, but I think by going through it, you'll remember much better its purpose. So here's a prototypical respiratory reductive dehalogenase. So I just popped up where you have hydrogen going to hydrogen ion in a free electron. This points out that the obligate dehalogenators require hydrogen. Without hydrogen, you have nothing. So the big blue ball there is a membrane-bound dehydrogenase. And the electrons that are lost from hydrogen in forming the hydrogen ion are transferred through the membrane-bound hydrogenase. You go through a series of molecules that you don't really need to worry about, and you go to the reductive dehalogenase, which are the red circles. Notice that vitamin B12 is central to that reductive dehydrogenase. And then as the chlorinated compound, which is represented by R-Cl, is reduced, you make HCl. And of course, that's disassociated in solution, so you have free hydrogens. Just as a side note, what happens to those free hydrogens? They go through an ATPase and they make ATP, which is the energy currency of the cell. So this is why we say that the halicoides respires chlorinated compounds. It reduces them, pulls the electrons off of them, concentrates the hydrogen ion outside the cell. When it pulls it in the cell, it makes energy. And that's how they live. But they can't live without the two key components, the membrane-bound hydrogenase and the red reductive dehalogenase. And key to each of these components is hydrogen for the membrane-bound hydrogenase. No source of hydrogen, you're done. And vitamin B12. No source of vitamin B12 to transfer the electrons from the cobalt to the chlorinated compound, you're done. So to emphasize that again, the reductive dehalogenase component requires hydrogen through the membrane-bound hydrogenase. And the reductive dehalogenase requires hydrogen, which came from the membrane-bound hydrogenase, and that cobalt factor, which is commonly known as B12. Without it, no membrane-bound hydrogenase were done, no hydrogen were done, and no reductive dehalogenase or the B12 factor, then we're done. So the central role of vitamin B12. In bacteria, there are probably 15 different variants of this cobalamin, this B12 that are made, and they have names. That's not important. We'll call it vitamin B12. It's the core ability of cobalt to form that metal carbon bond that allows the dehalogenation of the compounds of interest. As a side note, I'm aware that there are variability right here in the electron carriers, a lot of variability. Don't worry about it. It's not important relative to, it's important to the bacteria. It's just not important to you. It's not important relative to the things I just showed you. Now, this diagram emphasizes a little bit different point, and that is that the B12 for dehalocoides macarte and the obligatory dehalogenators must come from outside the cell. And the easiest way and the most sustainable way for it to come is if another bacteria produces it. So on the left here, kind of in the peach color, we have Sophospirillum multivorans. And this is a very common bacteria to see in the subsurface that can actually make the core cobalamin for the vitamin B12. It also so happens is a facultative dehalogenator. So with the PCEA, it can take PCE to DCE, but it cannot take it any further. And so it can respire PCE. On the other hand, if it runs out of PCE to respire, it can ferment starch or respire starch is another way of looking at it. And when it does that, it produces hydrogen and acetate. It produces more than that, but that's close enough. Now, the cobalium that it produces can either be remodeled, as in dehalocoides macarte strain BTF08, so it can change what's provided to it and then use it, or it can use it directly, as in dehalocoides macarte 195. The real core point is that if you can get these two working together, you now have the B12 that you need for the obligatory dehalogenator. It also kind of helps represent that you can, if with starch, some microbes can actually produce the hydrogen you need. So these are our facultative, halogenating, respiring bacteria, and obligatory. So I want to show you a chart that'll kind of help you think about that a little bit better. That's here. Now on the left-hand side of the chart, kind of flipped from what I previously showed you, is the obligatory organohalide respiring bacteria, of which dehalocoides macarte is probably the best known, though Dehalobacter is also well known. If you were to buy, say, KB1 would include a couple species, I shouldn't say a couple, a few species of Dehalocoides and Dehalobacter. These require hydrogen, as I've marked in yellow, but they will allow, in most cases, you to go from PCE all the way to ethene. The facultative organic halide respiring bacteria, which are on the right side of the dashed line, some of these are also well known. Geobacter is well known, right? Desulfibrio is well known. These can break down PCE and TCE, but can go no further. But the electron donors that it can use are much broader than hydrogen. As a side note, it is possible to have a site that is purely facultative dehalogenating microbes that will degrade the site, that it will degrade your compound. Okay. You don't see it on this graph, but it was published. Can't remember the person's name, but it was published probably 10, 15 years ago. But it's not common. So S multivorans is, as I showed you earlier, is on the side of the facultative. Now, obligate dehalogenating bacteria must have hydrogen. As I've mentioned, in starch, if you ferment starch, you can produce that hydrogen. In fact, Bacillus and Brevimonas together can exceed 400 milliliters of hydrogen per gram of biomass an hour. Plenty of hydrogen. The end products other than hydrogen are butyrate, acetate, and traces of ethanol. Most people just say acetate and move on with it, but you can produce the others, which is probably a different topic. The ratio of butyrate to acetate varies with other species. So if you take Cat 100 and it's starch and you ferment it, you get ethanol, propionate, formate, all of which, depending on the microbes that are present, go to acetate. And what we're interested in is hydrogen. So you can feed the obligate dihalocoides. Now I need to take just a moment to explain what next-gen sequencing is in the most simple manner. You essentially chop up the DNA. The DNA is then attached to a solid surface and you make lots and lots of copies. As you're making those copies, you're doing it with nucleotides that fluoresce. So you have a machine that can read that fluorescence as they're made. And then it's really the magic of being able to process large data sets that you put all those pieces back together like a puzzle. This allows you to see all the microbes that are in a sample. So here's the first data from next-gen sequencing that I'll show you. So this is Cat 100 microbial data, but this is only in the first quarter of a new emplacement. And notice the difference. On the left, I have the Cat 100, and I have the microbes from most prevalent down to least prevalent according to what was in Cat 100 sample. The one sample in the middle is the BOSS 100 sample. So remember, the Cat 100 is biotic, the BOSS 100 is abiotic. And then on the far right, I have a mixed impact control. Okay, so this is an area that had contamination in it but was not treated. So in the first quarter, notice the change. You have Acetobacter, Brevimonas, both of which can do some dechlorination. They're not all exciting, like the Dihalocoides example, but they're there. They also can make B12 and provide other helpful things such as amino acids and stuff. The only point I want to make with this picture is in the very first quarter, you make a fundamental change to the population that can clearly be seen. People always ask what the concentrations were on the site, so there you go. This was primarily a PCE site, excuse me. Okay, now this is another site. And what I want to show you about this site, having made the simple point that we can change what the microbial population looks like, is look at the controls we have. If you look up in what is the western part of the site, which is the upper left-hand corner, you see PDC 7, monitoring well 7. Down in the lower corner is 4, PDC 4. So these two are very far from any injection or the contamination. PDC 11 is an impacted control. It hasn't been treated, but it's close enough to the contamination that it's probably been impacted by it. PDC2 might be impacted by the injections. The injection areas will be represented by PDC 15 and 16. So they're right along on either sides of those railroad tracks. So plenty of controls. So what do the controls look like? Well, I could make a big deal and try to go through all these microbes with you, but we'll spare each other and just point out that you have a lot of microbes that are here, but none of them are obligatory dehalogenators. And in fact, remember, monitoring well 11 is in an area that's been impacted and not treated, and it's characterized entirely by almost entirely by methylotrophs. So methylotrophs are bacteria that can produce B12. That's good. And here's what the chemical contamination data looks like as a rule of thumb for this site. So there's no obvious dehalogenating bacteria. Notice the dates, 21 and 22. Okay, so this is the cat emplacement samples at monitoring well 15 and monitoring well 16 from 21 and from 22. Notice, boom, the Helicoides McCartae at 80% in 21. The Halobacter restrictus at almost 5%. You have the Halobacter further down and Geobacter lovely, etc. So the yellow Pac-Men represent known dehalogenating bacteria. The shaking hands represent metabolic support, something that's helpful to them. Say it may be making B12 or it may be making other things. Move to the next year at monitoring well 15, and you're at roughly 60% Helicoides McCartae, and you've kind of spread out the number of dehalogenating bacteria you have. Monitoring well 16 is similar to Helicoides McCartae at 95% of the population. It's almost unbelievable. You still have other degraders that are there in small percentages. You have a number of bacteria that can make the B12 that's required. And in 22, you see we've dropped to 71%, still a very, very, very high number. And you have plenty of support. This should impress you. Now, what I want you to notice here in this Bray-Curtis analysis is just simply showing you how different the controls are from the cat emplacement. It's something that's obvious to you, but this is just a different way of presenting the same information. And you can see that PDC11, the green ball, is very different than the unimpacted controls, both of which are greatly different than the cat emplacements. And the cat emplacements, even though they're very far apart, are very, very similar to one another. I would argue that we have control over the character of the aquifer post-cat injection, and that's why the two cat samples are so similar. Now, in 2023, we collected more data. I didn't have time to put it in the same format, but let's look at it. The 2021 monitoring well 16 data were 95% to Helicoides McCartae. We're 2.3 e to the 10th gene copies a liter. And 22, we've dropped down to 71, but we have other microbes present also. We're still at 1.24 times e to the 10th gene copies per liter. In 23, we've dropped to 44.65, still a very, very good number. And we're at 1.4 e to the 10th gene copies per liter. What you would see is that the microbial numbers in this particular case are staying very steady. But what's to account for the falling of the Helicoides McCartyi? Well, this is a graph that shows you the vinyl chloride and ethylene and DCE concentrations. I didn't need to show PCE and TCE because, as you recall, you wouldn't recall, I didn't show you, they're crashed. Okay, so they're gone. And we're really working on daughters at this point. And you notice that as vinyl chloride and DCE concentrations are falling, the obligatory Helicoides McCartyi, the one that has to have a chlorinated compound for respiration, it is also dropping. And that explains why you see its numbers. It's actually a confirmation that the site is being cleaned up. Okay, let's look at another site. This particular site is being driven by DHC. So it was stuck at daughters, and that's where we were called in to treat the site. You'll see at the very top our control. We had 7.6, 8 to the 10th gene copies a liter. But as you go through those, you don't really see any obligatory dehalogenating microbes. Maybe Geobacter is a facultative, that would be fair. But nonetheless, it's not a very exciting population. In the first year post Cat 100 injection, you see Helicoides McCartyi at 81%. Dehalobacter restrictus, basically 6%, the same with the other Dehalobacter TECB1 at approximately 6%. In the second year, you see basically the population replicated. Looks very, very, very similar, maybe slightly down on McCartyi and up on the other two. So Rectima, some people ask about that. That's a big hydrogen producer, and it can produce it if you give it mono, di, and polysaccharides, which is exactly what you do when you give it starch. Pseudomonas florensis, it has the capacity to produce B12. So we have the core elements we need to be successful. Now I want to show you kind of a larger, I stopped showing you what the microbes were at 0.4% in that second year. So I opened it up to show you to the bottom of the list. And why did I do that? Well, because these are a bunch of potential or actual producers of B12. If I've said it's potential on Streptococcus and Clostridium, it's because I don't know what the species is on those particular samples. So what are my observations from this site? That obligate dehalogenating bacteria have arisen and increase in abundance. The obligates are needed to reduce DCE because we're on that other side, right? The source of hydrogen is present. Vitamin B12 is present. There's an initial reduction in diversity. You see that sometimes, usually with some specialization like dehalogenation, right? And then there's an increase in total abundance over time. We're not seeing that yet. You do see it in the second year versus the first year, right? Give it more time and you'll see it increase more and more until you get to the end of remediation, in which point you see that the dehalogenators disappear and initial or natural abundance increases. So diversity and abundance increase as chlorinated ethenes decrease. And I'm circling Clostridium there. It's a very interesting microbe, and later on I'll give you I'll offer a presentation on why that's important. But now I'm just going to leave it as a bug in your head. Site 4. So this is a PCE-driven site with ppm concentrations, but lower ppm concentrations. And this data is presented in a little different manner. And what you need to kind of have an understanding of to make sense of this is that in some cases I'm going family, genus, order, whatever it is, to emphasize that you're not seeing any Dihalocoides. So for Dihalocoides, the phylum, and it's in the middle of the graph here in blue, the phylum is Chloroflexia, the class is Dihalocoidia, the order is Dihalocoideales, and the family is Dihalocoideaceae, and the genus is Dihalocoides. So do you see any Dihala anything in the pre-treatment on the far left in 2018? You don't. Now, there are a number of bacteria that are doing interesting things. So the Methylophiliacea, which is at the very top of the chart, and the Oxalobacteriacea, which is also at the top of the chart, being 31 and 20% respectively. These are pretty interesting. They're using nitrogen to work on probably alcohols, but has nothing to do with PCE. You have some biosurfactants. You have some that can degrade alkenes. Very interesting. They're very busy guys, but they're not busy with PCE. 45 days post CAT 100 treatment. That's the column in the middle. And what do you see? You see Geobacter raising up. You see sedimentary bacteria raising up, both of which are facultative dechlorinators, but not particularly excitable. Depends on which bacteria, Geobacter species we're talking about. But you can see some changes, right? And that would be consistent with what I showed you very early on in the presentation, that the CAT 100 injection fundamentally changes the microbes that you're seeing, even before you see the rise of Dihalocoides. Now, if we move to the right, we see the saturated zone 2 years and 4 months post-treatment with CAT 100. And you see Dihalocoides McCarty at 41%. You see some other microbes, especially down in the low percentages, that are also dehalogenating microbes. Now, this site is not as exciting. One of the reasons being is its overall concentrations are low, much lower than the site where I showed you that you had 95% Dihalocoides. So this is about where you would expect a site like this to be, because it needs the chlorinated compound. It respires it. It has to have it, just like it has to have hydrogen, just like it has to have B12. So as your chlorinated compounds go down, the Dihalocoides will naturally follow. I've also marked some archaea here, some Methylosarcea. They can also be B12 producers and things like that. So what are the observations that we've made in this kind of little voyage through some data? On sites that seem to lack Dihalocoides, based on next-gen sequencing, which should be sequencing basically everything, all the DNA it can find, the installation of CAT 100 is followed by Dihalocoides that range from 40 to 95% of the microbial population. This is without Dihalocoides augmentation. In the first quarter, the microbial populations post-BOS 100 change the microbial characteristic of the aquifer and show varied metabolic pathways. What's required? Vitamin B12, cobalamines are required by obligatory dehalogenating bacteria. I probably didn't say it, but I'll point it out now in case one wonders. The reason I'm making a big deal about the obligatory dehalogenating bacteria is if they are there, then they are dehalogenating because they have to. That's why they're more important than the facultative. The facultative could come and go and be doing other things for a living. After the CAT 100 installation, the number of bacteria and archaea that can produce B12 increases. Bacteria that ferment sugars to hydrogen also increase. As the available halogens decrease, the DHC population follows suit. As the DHC population decreases, the microbial diversity rebounds, heading back to the natural state of the site. So that's been our little voyage through a little data today, and we'll see what people have as far as questions go. Derek, it's your turn.
Thanks, Ed. If anybody has any questions, we have both the chat and the Q&A open. Feel free to drop those in there. There was one that popped up here a second ago, Ed. I think it's related to the slide you have on the screen. And the question was, are there other bacteria, either indigenous or that are added to some of these mixes that suppress microbial diversity besides DHC?
Yeah, I do wonder. In KB1, they add some methanogens, which are good for producing B12, but I can't really understand why you would want to do that. First of all, they're available in the subsurface most of the time if the environment's right, so I wouldn't bother. I think it's historic more than anything. The other problem is that if, and I don't think this is a big problem, but I'm throwing it out, if you build up too much methane, you start suppressing other metabolic pathways. So, there are bacteria that could suppress various you can suppress the population, but DHC is such a specialized microbe. It's not surprising that it would kind of suppress things going on around it as long as it's getting the vitamins, the B12 and the hydrogen and things like that it needs. It's very small. It's very specialized. It's very dominant. And there's also quite a bit of energy to be harvested by dechlorination, at least for those microbes. So I don't know if that really answers the question, but it's so different than, say, petroleum sites where, post treatment, depending on what you're treating with, but certainly post treatment of, say, a BOS 200 plus, you will actually see diversity go way up. But on chlorinated sites, you tend to see it focus down.
Gotcha. And then the follow-up on that was, are you inoculating with B12 or how are you delivering B12 with the Cat 100?
Okay. So it's a combination of things. Yes, we put yeast extract in, which would provide B12. And so, yeah, we start off with B12 to begin with. But after that, what you see is that microbes that produce B12 also flourish, and they're primarily flourishing off the starch would be what I would expect.
Okay. And another one here. Are you essentially using the activated carbon of the BOS 100 to compress an EISB timeline? EISB being enhanced in situ bio.
Well, it would be hard to say no. So I think the answer is yes. And that probably invites more elaboration. So the iron is inside the BOS 100, for which cat is the extra additives make it cat. So you're getting abiotic degradation within the interior of the BOS 100. And with that, you also still have the fundamental characteristics of activated carbon, which tend to increase to some degree enzymatic reactions that are close to its surface, that are involved in other microbes that react with surfaces. It also makes a very good place for microbes to start building communities. And quite frankly, that as I showed you early on, that you would see the interaction with the helicoides and other microbes that provide the things they need. So this would be the kind of community you'd want to build. It's well published throughout the literature that activated carbon is very good for building biofilms and microbial communities. So I hope that answers the question, but I think so.
Sure. Another one here pertains to geology. Are there geologic conditions such as low pH, salinity, or high clay content that could affect the efficacy of the Cat 100 or the robustness of the population of microbes?
Yeah, it's hard to say no to a question like that because the um, geological variables can range so greatly. But generally within pH, say a pH of say 6 to about 9.5, you probably don't have much to worry about. If you start getting outside those pHs, you could start having issues. Clays, really our products do really well in clays because of the way they're installed, the way that we break up the clays. But regardless of how great our products are for clays are an inherent problem in distribution, right? So I couldn't say no, that's not important. It would be. Let's see, what was the other point, Derek? pH, clays.
pH and salinity.
Oh, salinity. Yeah, that's kind of the same too. It's almost like, well, what's salinity, right? So a lot of the dehalogenating microbes are found in the deep ocean, so they can handle salinity. We would have to look and see. It'd be nice to see a site where I had actual data from it, so I don't just have to pontificate from nothing. My guess is they're pretty robust against salinity, but I do not know that.
Okay. At this time, I don't see any other questions in the chat. So if someone has a burning question, feel free to drop it in now. We'll give you a few seconds here. While we're doing that, Ed, I just wanted to thank you for your time. This was very informative and I found it interesting. So I hope others did as well.
Well, good. I appreciate it. I hope everyone enjoyed it. And I appreciate also each of you being a guinea pig for what is a new presentation. If you see something in the presentation you thought was confusing, unclear, or should be emphasized or built upon, just drop me an email. I'd appreciate your help.
Sure. I appreciate you offering that up. The slides was a question that can be offered up as well, and this will also be posted as a video on the RPI website and YouTube channel as well. And then Ed, just if you can't see the chat right now, there's a couple people saying thank you for the presentation. You made a complicated subject easy to understand.
Oh, that's very kind.
All right, so we have no other questions. We'll conclude there. Give you back about 5 minutes of your lunchtime. Thank you very much, everybody, for joining us. And if you have any questions, please feel free to directly reach out to Ed. Thank you.